and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to two researchers, Viau and Dr. Adrian Paul, one of whom currently works and the other formerly worked under the direction of Michel Lebrecq, who lead a group focusing on phytoremediation at the Plant Biology Research Institute in Montreal. Contamination of various kinds can make it difficult to ensure healthy and safe food crops in urban agriculture. Phytoremediation is a strategy where we may be able to use plants' incredible natural abilities to help with this problem. This episode, Marianne and Adrian join me to discuss harnessing phytoremediation to help clean soils for tomato crops. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Marianne and Adrian with us. Marianne Viau is a biologist who has been active in urban agriculture for 14 years and sees it as a way to bring nature back to the city. She decided to dedicate her master's degree at the Plant Biology Research Institute in Montreal, Quebec to one of the obstacles of urban farms, contaminated soils. She's now a teacher at Collège Antique, where she is tasked with developing a technical program in urban agriculture. Originally from France, Adrian Paul is a young ecologist with a PhD from the University of Queensland, Australia, where he focused on understanding metal-enriched ecosystems. Now postdoctoral fellow at the Plant Biology Research Institute in Montreal, Quebec, he's shifting his focus to phytoremediation, developing innovative solutions for urban land remediation. Adrian has published over a dozen peer-reviewed articles demonstrating his commitment to addressing environmental challenges. Hello, Marianne and Adrian. How are you both doing today?
Hello. Perfectly fine.
Hello. Good. And you?
Good. So good to have you. Apologies to the listeners. I'm getting over a little bit of a cold, so if I sound a little different, my apologies. So today we are talking about phytoremediation and fixing contaminated soils in urban settings. So to get us started, can you just kind of provide an intro to what that term means and how it relates to the problem you're trying to address and just kind of the problem in general?
Yeah, but for the problem in general, as you were saying, I'm working in urban agriculture for more than a decade. So for everyone to understand, urban farming isn't just about planting seeds in the ground in the city limits. It's more a complex cycle that includes growing, selling, processing, and even reusing plant waste. This approach to farming doesn't rely on the single crop strategies typical of conventional agriculture. Instead, most of the urban gardens celebrate diversity, mixing different plant species to produce multiple harvests each year. This multi-cropping approach serves a dual purpose. It naturally fades off pests and improves soil fertility. And it's not a new concept at all. We use a concept that were used, for example, by First Nation in the Great Lakes area predate the European colonization. And now, more in the 21st century, urban agriculture faces a significant challenge with soil contamination that pose a risk to food safety and community gardening initiative. However, the level of risk to crop varies and is influenced by multiple factors such as the pH of the soil, type of contaminant, and the plant species, like usually what we eat in the plants. For example, if we have leafy vegetables such as lettuce, well, we eat the leaf and unfortunately, usually the leaf absorb higher level of metals, so that can be less sustainable to grow lettuce if you think you have a contaminated soil. So in the light of all these challenges, our research focused on the synergy between a food crop that is the cherry tomatoes and companion plants, which are known for their ability to detoxify soil or fertilize them.
Sure. And just so people understand the term, phytoremediation, that's that process of like using other plants to clean the soil?
So yeah, I will talk about that. Phytoremediation is actually a green technology that uses the natural capacity of plants to deal with contaminants. So globally, it involves their use to remove, stabilize, transfer, or degrade contaminants from soil and groundwater. To be fair, that's like, if I can go a bit further, this technology is actually very polyvalent, and it will include a range of strategies depending on the type of contaminant. So we have different types of contaminants: organic, which is basically like hydrocarbons, so basically crude oil, coal, and so on, and inorganic metals mostly. I have a few examples for you. Some plants can stimulate the degradation of these organic pollutants, such as polycyclic aromatic hydrocarbons, which is a class of chemicals that occur naturally in coal, crude oil, or gasoline. Other plants can stabilize non-organic toxic elements, including metals like lead or copper that Marianne focused on, and prevent them from spreading into the surrounding ecosystems. They will do that through different ways. One is called phytostabilization, where plants will trap these contaminants in the root zone using chemical compensation, among others. Another one that we actually use a lot in Montreal, Quebec, is phytoextraction. And this one includes the uptake of these contaminants inside the plant system and the transfer from the roots to the leaves and the stem of these contaminants. That's like a pretty detailed summary, I guess. I could go further if you want to.
I mean, feel free. I think it's super fascinating.
That's fine. I can go for the advantages unless like you have several other questions or like you plan to ask this question. But this phytoremediation has like many advantages, also face challenges. I always like to finish with advantages for the people to keep that in mind, but I'll go with the advantages first because Marianne is trying to assess and not trying to like find a solution to the challenges. So you have to know that in Montreal, we are focusing on phytoremediation in urban areas. As she said, we focused on a place on the eastern part of the island, because Montreal is an island actually, if you didn't know. And we focused on the use of trees, which are like willows and poplars, to extract, stabilize, degrade the contaminants, as I said, but also to promote biodiversity. That's something I will discuss later. I know that you are very interested to discuss about that. I've seen some of your questions. You have to know also that like phytoremediation may provide social benefits, so I will be able to discuss that later. But the main point also is it's very cost effective. You have to know that like most of the case, cities and like stakeholders will decide to use a technique that is called dig and dump. So basically, they will dig, remove the soil, put it somewhere else, and put like a cleaner soil inside. That's very expensive because it's like requiring a lot of equipment, a lot of people, and so on. So using plants might be a bit slower in time, because it's not done in like a week or in 2 weeks. It will take years. But in terms of like cost effectiveness is a lot better. The last one is obviously like the low environmental impact with reduced carbon footprint, no use of chemicals, and the fact that it's blending perfectly into the landscape. So that's quite a few advantages. Could go for the challenges, but maybe you want to ask questions later.
Yeah, I love this because there are many things I love about this research. One of them is that I love that you're just kind of partnering with things that nature can already do to solve the problem. And we've had other episodes about remediation of different kinds of soils and from different types of contamination. And yeah, I mean, it can be really expensive if you're moving soil or just it ends up in a landfill or something like that. Like, it's not very helpful in certain ways, obviously really helpful in other ways. So it's nice to be able to see kind of an alternative option that with a little patience is going to pay off in some big ways. So you've lightly touched on this already, but you are looking at kind of using some of these crops to help clean some soil for tomato plants. So can you talk a little bit more about kind of what you were hoping to test, or what you were hoping for your hypothesis in this research?
Yes, absolutely. So as Adrian was saying, there's an interest that is growing around phytoremediation. And so it was interesting to mix it with food crops for the contamination of the land and the use for urban agriculture. So, and we try to identify some safer methods because, it's still something that is related to our health. So we have to be cautious about it. So the current research focus on preventing the translocation of the contaminant, especially copper, in the food chain, and try to do it with strategy also used in urban agriculture. As I was saying, the diversity of cropping, so the companionship between different plants. So this study investigated the efficacy of using the food crop, that is the cherry tomatoes, with the combination of several companion plants that can remove soil contaminants and improve the soil quality. So our primary objective was to advance the scientific knowledge to develop more effective approaches to remediate contaminated site and have safe production at the end too. So the plant that we choose as a companion with the cherry tomatoes have phytoremediation properties. So first of all, we choose the pussy willow. So as Adrian was saying, willows are really used a lot in phytoremediation research as a shrub, a tree that is really efficient for removing contaminants. We choose also the common yarrow, that is more an herbaceous plant that is also really present naturally in brown fields because it's a resilient plant. And there was a little bit of research that was talking about its phytoremediation properties. So I was interested about it, and also they produce beautiful white flowers that bees love. So when we think about urban agriculture, we also think about urban beekeeping. It's really, pollination is really important if you want to have a good food production. So when we think about the food chains, I was also thinking about, the pollinator, can they have problem with the flowers if we have plants that have property to take the contaminant? And in the same ways, we think about the white clover. So I hope everybody that is listening tastes honey with clover because it's delicious. But also it's a really good green crop. We use it as a naturally green fertilizer. So we want to use it like this to help produce nitrogen for our production. So our hypothesis was that the tomatoes that were growing with the willows and the clover together were gonna have less contaminants in their fruits versus the rest of the companionship that we try.
Nice. I have so many things off of that I just adore. I'm really glad that you mentioned the bees and kind of the question of, if they're getting pollen from plants that are taking up these contaminants, are they going to be cool? So I have a question about this before we move on. For things like a pussy willow or yarrow or whatever that are kind of more in my mind, and I could just be confusing my willows here. Is that like you said it's kind of a shrub? Is that like a woody perennial kind of thing? Okay, good.
Yes, both are perennial plants in Montreal, but the yarrow is more herbaceous. It's not wooden at all, but the willow is.
Sure. So then when you're growing these side by side, how is the tomato plant, like, what does that physically look like in the sense of like, I like my mind goes to like, okay, if you're planting like a tomato plant like right next to like a raspberry bush or something, which is like an easier visual for me to conceptualize. Like, how is the tomato not just like overtaken by this potentially much larger plant?
Well, that was one of my thoughts when we think about this research at the base, is that, well, in nature there's traits. In the forest we have the big trees, then we have the shrub, then we have the herbaceous, and together they find all their ways. And in more permaculture ways to do urban agriculture, we try to do the same. But of course, there's also the fear that they're gonna be in competition, and that was also one of my fear too. So, but we want to try it if plants that are in the same traits of both herbaceous plants, tomatoes and yarrows, they are in different traits. So, willows and tomatoes, if they're gonna have a difference. And of course we didn't plant a mature willows, it was cutting, and it was seedlings of tomatoes, so they grew together, so it was not so bad as a competition. But of course, one of the reasons why we love willows in phytoremediation is because they grow fast. So of course, at the end of the experience, the willows were much higher than the tomatoes. But tomatoes can go really high too, if you think about in a big production in greenhouse. So it can be equivalent in a way. And of course, for the research, it was really a beginning of experimentation for my master. So we put them in a container. So it was really urban agriculture way, but in a vision for the future, and we can talk about it more later, but it could not be in the same container, but in the land where there's, a big field with the tomatoes in the middle and maybe the willows on the side. So the competition gonna be less present, but we want to just see, first of all, the interaction between both of them before going forward into that.
Sure. Great. So you've touched a little bit on kind of the setup physically for this, which leads really well, I think, into our next set of questions, which is really about the methods of how you went about this research. So I'm curious as to just kind of not only what kind of setting you were doing this experiment in, because obviously you can have anything from like an urban, like multi-layer food garden to just someone planting tomatoes on their balcony, all kinds of things. I'm curious where you're getting this contaminated soil from, or how it gets contaminated in these settings, what you were doing your research in a greenhouse, what kind of, or whatever setting you were doing that in, as well as just how you were testing these various mixes that you had going.
Okay, so basically, like, the soil that we used was coming from a site that is located on the eastern part of the island of Montreal. It was like, we have 4 hectares on this part of the island, but the site that Marianne worked on is a 2-hectare site, or 5 acres for the American listeners. And so it's located in a densely industrialized area of Montreal, surrounded by numerous petrochemical and metallurgical plants. It was originally used as a snow dump, but the site had become a brownfield before we began our research in 2016. So basically, it was often used for illegal dumping by businesses and individuals seeking to avoid waste disposal fees. But today, the site contains a diverse mix of waste, including physical debris, such as construction materials and metal, as well as organic contaminants and heavy metals, such as copper, selenium, lead, and manganese. The main one is copper. That's why Marianne focused on this one. You have to know also that, like, we have on the very specific part of the site quite a high concentration of selenium. Regarding the level of contamination of the site, in the province of Quebec, where we work, contamination levels are categorized using letters, with C indicating that decontamination is required for any reuse of the site. B means that, like, above this level, we cannot use it for, like, residential use, but we can still use it for industrial use. So basically, like, on this site, many areas are above C, meaning that we need remediation. Like, we absolutely need it. We cannot do anything besides remediating before industrial or residential repurposing. I can let you continue, Marianne, on the actual methodology.
Yes, of course. So the experiment was conducted over a single growing season. So in Montreal, it's around 4 months. And during several key parameters were monitored, like the plant growth, the fruit production. So we count a lot of fruits and flowers also during the season. We, of course, tried to monitor the best as we could the copper concentration in different parts of the plant. So the fruits, the stem, the leaf. And we also make an overall effectiveness of the polyculture assemblage in reducing the soil copper levels, so by containers. So if we do more a graphic picture of it, we can imagine that we try to formally transform a contaminated brown field into a green oasis where there was tomatoes and a lot of beautiful green plants that were together to revive themselves and also make good food. So it was in the open air greenhouse in the Montreal Botanical Garden in Quebec. So this is where also our Plant Biology Institute's research center is. And so in the greenhouse we put 60 containers to grow our 5 plant companionship together and 3 types of different soils that we replicate in 4 times. So it was 4 blocks with 15 containers by block. In every blocks we randomly put our different polyculture. So there were 4 polyculture and 1 monoculture of tomatoes. So we want to see just tomato alone, what can happen. Then we put also 2 plants so it was 3 plants of tomato for the monoculture. So 3 plants by container, 1 with just tomatoes, 1 with 2 plants of tomatoes and 1 plant of willow, 2 plants of tomato and 1 plant of yarrow, 1 plant of tomato, 1 plant of willow and clover that cover the ground, and finally 1 plant of tomato, 1 plant of yarrow and clover that cover the ground. The 3 type of soil were Adrian was explaining earlier. So we take the techno soil from the brown fields in the industrial area of the east of the city of Montreal. And this is where we have experimentation with the laboratory for several years. So the copper, because there's phytoremediation there already, is around 100 milligram per kilogram of copper. Then we use the same soil, but we spike it as is original higher concentration, so around 2,000 milligram of kilogram of copper. And finally, our control group of soil was a typical garden soil to see also just how the plants interact in a healthy soil, also if there's a difference in the production too.
Sure. You've mentioned already that different types of plants, kind of when they interact with these contaminants, can put them in different areas. So some of them have them trapped in the root zone, some of them are taking them up into the leaves. Are tomatoes specifically susceptible to this, specifically copper? And where do tomato plants put that? Do they put it in the fruits? Do they put it in the leaves? What does that look like?
Yes, but I can start, and if you want, you can complete, Adrian. As I was saying earlier, different plants of different kind of plants, species, gonna have contaminate in different place. I was saying, leaf and roots, especially roots, gonna have a lot of the contaminate because, logically, they are more around the ground, so it's easier to grab it. But why we choose fruits plants is because the studies shows that usually the fruits, the plants try to protect it, because where there's fruits, there's seeds. And where there's seeds is the new generation that the plants want to, protect. it's their babies, so they don't want them to be contaminated. So usually there's less contaminants in the fruits. The plant's gonna exclude the contaminants from the fruits and put it more in the leaf, the stem, and the roots.
Yeah, but you also, if I can continue on that. It will be very species dependent and family dependent. You will have when I was working in Australia, I was focusing on the accumulation of nickel, which can be another contaminant, not in the case of Montreal. But you have plants that are capable of hyper-accumulating these metals, meaning that for the case of nickel, they can accumulate up to above 1,000 milligram per kilo. I will just use ppm because that's the same as milligram per kilo. Above 1,000 ppm. But I was actually working in New Caledonia on a plant that could accumulate up to 25% of nickel. So it has a green sap. Basically, that's literally the color of nickel that you can see on this plant. For people, if they want to check on Google, it's called Pitnandra acuminata. And it's pretty impressive. Coming back to hyperaccumulation, plants can hyperaccumulate quite a few elements, like nickel is the main one, cobalt could be one, copper, some of them, and selenium and so on. In North America, the main one will be selenium, especially around the Great Plains, like Colorado and so on, going up to Saskatchewan for the Canadian. So basically we have these plants, but sadly we don't have many where we work in Montreal that are native. So that's why we have to use willows and poplars. So willows and poplars are not hyperaccumulators, but they may be considered as accumulators. So they can have pretty good concentration within their tissue. But the huge advantage is that they grow fast. So it's better to have let's say I'm saying something very rough, but it's better to have a 50 kilos tree that gets 10 ppm or 10 milligram per kilo than having a plant that is 5 grams but get 1,000 ppm. You remove more from the soil. So that's the idea also of using willow and salids, willow and poplars. But coming back to the tomatoes, tomatoes are actually, these kind of plants, fruits won't have much. Very, very low concentration, unlike leafy vegetables like lettuce or spinach. I actually did a study in California focusing on spinach that was grown on soil that was pretty concentrated in titanium and can be a problem. So actually farmers are and scientists are trying to find solutions to limit the uptake of this titanium into the parts that we actually eat, the edible part of this lettuce and spinach.
Fascinating. Yeah, I think for me, non-scientist, as everyone knows, I would have assumed that, because they're putting so much of the water and nutrients that they're getting into growing this fruit, that there might be some kind of just throw everything we get in there, whatever it is. So it's very cool that the plants are able to have that level of discernment, I guess, in what gets into the plant. And I know there's multiple mechanisms. It's not necessarily a sentience thing there, but I think that's really fascinating. Hi everyone, I hope you're enjoying the show. Interested in learning more? Marianne and Adrian's paper, Can We Simultaneously Decontaminate and Cultivate an Urban Cherry Tomato Story, published in Urban Agriculture and Regional Food Systems, is always freely available. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. So, then what did you find? What were your results?
Well, the results largely support the initial hypothesis that it's feasible to simultaneously decontaminate soil and cultivate crops in an urban setting through carefully designed polyculture system. So what we saw is that certain crop combination like tomatoes, white clover, and pussy willow together not only thrive the contaminated soil full of copper, but also produce a substantial yield of tomatoes that were mostly safe as a level of copper. So that's why it's really hopeful to make more research about it and maybe one day change our brownfields to urban farms.
I mean, if I can continue. In the field of phytoextraction per se, the study underscored the potential of using polyculture systems in phytoremediation to enhance biomass production and contaminant removal from soil. So the integration of these specific flora, such as yarrow and clover, into this system significantly influenced both yield, like Marianne said, but efficiency of copper extraction without increasing Cu in fruits. In the case of clover, its incorporation enriched the soil with atmospheric nitrogen. That's basically like, I mean, people might not know or know, but legumes are capable of stimulating the integration of nitrogen into the soil through like their association with microorganisms. And so like this phenomenon actually promoted the overall growth of all the plants that were associated with this legume. For the yarrow, it's very resilient and actually require little water. And so it was not competing per se with the tomato, which actually requires a lot of water, especially during the growing phase. And you have to also know that during the growing phase, it's actually pretty common to have like heat waves. So like water is kind of like something that is crucial, and having this plant that doesn't require that much water is very interesting, especially since, like Marianne said, yarrow is actually also pretty well known. Some studies actually showed that it was very good at stabilizing copper in its roots. So taking advantage of this low water requirement and capacity to stabilize copper was a pretty interesting thing in the case of yarrow. Yeah.
I have a question off of this because I know you mentioned it in your paper of how you actually then go from having these contaminants drawn up into the plants or the root system to actually then getting it fully out of the soil. Because presumably, if a plant takes up a bunch of copper and then just dies and then decomposes in that soil, it's going to go right back into it. Mm-hmm. So can you talk to me a little bit more about, I know specifically you handled this in your project with some kind of leaf collecting, but how, I guess how you did that in this project, as well as how that might apply on like a larger scale.
In the case of Marianne, everything was in containers in a greenhouse, so we didn't have this problem to deal with. As you said, in our project, so we have like quite a few projects all in Quebec, but all over Quebec. And what we do is for like the leaf litter, we just like collect it and like put it somewhere for like further use, which basically will be like sent or shipped to centers to burn it to recover the to recover the energy. And obviously, like these centers have systems in place to like collect all the dust that is enriched in metals, and this dust will be actually like sent to dump sites. But what you have to understand is that the metals have been removed from the soil, and then we reduce the size, like the actual size of the contamination from like 50-80 kilos of trees to like 10 grams of dust or whatever. It's not like the good ratio, obviously, but that's the idea. I'm getting lost in my thought, but yeah, like that's the idea. It's to collect these these dusts, to like dump them, and then we also have a process of copicing. So every 2-3 years for like willows, we'll copy them, means that we'll cut them 10 centimeters above the ground, and then that will stimulate the growth, and this way we also remove the contaminants. It's not staying in the field. Like this biomass will again be sent to these burning facilities to produce energy and then dust will be sorted. So like we have two ways of controlling that.
Yeah, I found that to be such like a charming aspect of your research, of just like collecting these leaves and like what a smart way to remove that contamination. Is it's just like little packages gifted to you by the plant of please recycle this. So
Yeah.
I really like that. I have a point on here for just kind of some of the broader implications for this research that you got from your results. So, Marianne, did you want to talk about that a little bit?
Yes, sure. Well, as I was saying, the results of this study highlight the significant opportunity for urban agriculture to contribute more to the food security of the city if we can have access to other lands that for now are too much contaminated for using them and still have a good health at the end of the production. So, and what I like about this also is to put more in front, of people that polyculture still are a system that could be efficient and could produce a lot, because we are more in a system that produce a lot for monoculture, that is seeing like the more efficient and productive way to produce. But as we were looking in this study, if we put together plants that can be helpful to each other, at the end we can be surprised as the production can be higher than the monoculture itself and healthier. So it's really one of the main result that is interesting, and that we want to provide more practical recommendation for farmers and urban planners to advocate the intention of multifunction in the green space. You know, that in a landscape that we are not sure of the contamination, or we know that there's contaminants inside, well, at the place of all dig and dump, as Adrian was saying, well, we can put a part of it in phytoremediation, and slowly but surely, after a little bit less than a decade, we can try to produce more fruits, plants, and be able to have a little production and be okay for our health and for nourishing our community. So, of course, we have to do more research about it, but I hope that's gonna encourage further interdisciplinary collaboration to see the full potential of polyculture in different contexts.
Sure. Yeah, and that's a great lead into the next question, which is actually you have multiple areas of future research. So what does that look like?
To be fair, like with Marianne's studies has not yet led to further investigation in this area, but a forthcoming student-led follow-up study will focus on assessing the oral bioaccessibility of contaminants found in vegetables and their surrounding soils in the context of phytoremediation project like the one we discussed about. I know that you were interested to know if we're like about to work on other contaminants. Copper is the main one for this site. We could actually work. One that interests me a lot is selenium, as I said, because we have a part of this site that is very highly contaminated with selenium. So we did some work on this selenium, and I would like to include the work that Marianne did, or like adapt it in a way that it's done in the context of selenium accumulation. But if I have to be a bit more general in terms of future research on the ecosystem services provided by the combination of urban agriculture and phytoremediation, as you know, we already do this work about soil health, which is like a very important part of ecosystem services, through like the reduction of contamination and enriching soil fertility. However, like there are like many areas that we could focus on. Actually, just before this interview, I read a paper by Werther Guidi, which is a former collaborator of our group in Montreal. He actually returned to Milan now, where he's from. But like he actually wrote a nice paper about how a multidisciplinary approach could like enhance the understanding of all the processes that are actually occurring into like phytoremediation, but also for like urban farming. We could actually focus on supporting biodiversity, especially local wildlife, on improving local food production, contributing to food security, facilitate climate regulation, providing social and economic benefits such as fostering community engagement and strengthening local economies, improving aesthetic and mental health through enhanced environmental quality. So this kind of research would include let's say art science, like Marianne and I, or people that are more into social science, which is something that sometimes in our field is kind of not regarded as much as it should. But that's something that I would like to start with the collaboration of Michel Labrèche, which we should have said his name before that, but he's the leader of our group. And even at the RBV, or a plant, in English it's Plant Biology Research Institute, we could actually try to integrate social scientists. That would be actually great and something I'd like to do in the future. But in the end, the ultimate goal would be to highlight the multiple benefits of combining these two technologies or these two strategies that are phytoremediation, which started a couple, 2 or 3 decades ago, at least in Montreal. And urban agriculture, which is very big, becoming very big, and Marianne is actually contributing to that because she's part of organizations. I know that she's very much involved, so that will be great to meet the two of them and make the future brighter.
Yeah. Oh, I love that. I'm such a huge fan of multidisciplinary research and just how everything is so interconnected. So that's what a lovely note to end most of the interview on. But I still have 3 questions left for you. So the first one is, if people want to learn more about any of the things that we've talked about, where can they go to do that? And also, Adrian, if you send me a link to that paper you mentioned, I would be happy to include it in the show notes. So other than that, what can people do to learn more?
Well, if they want to know more about urban agriculture and also all the struggle that we have with contaminated soil, there's 2 books that I pretty love, but they're kind of exhaustive and advanced, so you have to be a little bit motivated if you want to. But still, there's the one that written by Ackerman in 2012. That is the potential for urban agriculture in New York City. So the growing capacity, food security, and green infrastructure. And they have a beautiful chapter about soil contamination. So, of course, it's about New York, but it's one of the big cities of North America. So it's always good to read about it. And the other one, it's Carrot City. That is more a collection of ideas and there is that talk about, yes, food production, but also more design, urban design, and talk about a lot of different cities. So there's Toronto, there's Montreal, there's also more American cities. So it's really interesting. So it's on the website of the University of Toronto. So it's a really interesting book and articles too. Of course, there's a lot of video on YouTube about urban agriculture in Montreal if you want to know more about it. Unfortunately, most of them are in French, but some of them have subtitle in English. Also, if you want to know more about the research that our main researcher, Michel Labrèche, is doing, there's still a lot of news. It's kind of popular on the media, so you can find most of them also, but especially in French, but there's some of them in English. That we're gonna have put some link about it.
Yeah, I could actually send you some links for the English part, but for the French Canadians, he actually made a documentary on La Semaine Verte, so that's a very nice one. Other than that, I know that some English media actually contacted him, so I can actually share with you all the links and so on for all this. For everybody that speaks English around there.
Perfect. Wonderful. Yeah, we'll include a bunch of links in the show notes to that kind of content. So then, second question is, if people want to take the next step and kind of look at getting involved in this kind of thing, how can they do that?
Yes, well, For listener, I don't know, even where you are, usually there's maybe a little bit of urban agriculture. So if you have a local urban farm, but also just all the little farm nearby your city, just to support them, to buy local, to support the producer. It's a thing that I think it's really important to do right now, to improve our food system. In Montreal, we have a lot of bigger groups that represent a lot of members in urban agriculture. So we have Cultivation, we have Cultiver Montreal, we have what we name iLab, that is the laboratory of urban agriculture in Montreal. So yeah, we just have an article from them that examine 10 cities of the world, and we have the one with the most urban farm by habitants. So we are really proud of it. So yeah, that's it. But of course, also if you in a more political way, it's also good to reach out our local representative and city council member to have more policies and programs to promote safe food, urban agriculture, but also to manage our soil health that is not protect enough in the cities and around it. So in Montreal, we have our first strategy about urban agriculture, and we work hard to create more food policies. So we hope that most of the city can join the movement about it.
Great. Those are wonderful ways for people to get involved. So then, final question for you is, what is one fun fact for each of you that listeners wouldn't know if all they had was your research?
Go for it. That's a very tough question right now. I actually thought about it in a way. I spend a lot of time in my lab, in my office, doing stuff. The one thing that I love is I'm also a huge fan of sports, so I decided to get ready to run the Marathon of Montreal next September. So that's something I've been working a lot, but sadly I'm injured right now. So I'll try to do my best and recover ASAP and get ready for, I think it's late September. So I'll try to get ready for late September.
Wow, that's amazing. Congratulations. Yeah. I hope you recover in time.
Yeah.
Yeah, for my case, I'm a really proud cyclist that try to be most active in the four season. That's kind of our core in Montreal because it's really winter biking also that is really cold and snowy. But so it's a marginal practice, but it's more and more popular in the city every year. So it's really fun to watch, and it's good for health, for the environment, and it's fun. So I'm proud of that.
Awesome. Well, geez, all you need is a really strong swimmer and you'll have a really strong triathlon team there.
Yeah, it's true.
Well, this has been such a lovely conversation. I adore this research. I adore what you guys are doing. So thank you so much for your time and coming on the show today.
Thank you so much for the invitation.
Thank you so much.
Hi, everyone, and welcome to our Student Spotlight, where we highlight the work of graduate and undergraduate society student members. Today, we'll be talking to Transcripts. Welcome to the show. Can you start us off by introducing yourself and where you're studying?
Yeah, sure. First of all, thank you very much for having me on the show. As you mentioned, my name, I am Transcripts. I also go by Charan, and I am a PhD candidate in Plant and Soil Science Department at University of Delaware in Newark, Delaware.
Awesome. And what are you currently researching?
So I'm studying plant-microbe interactions. So my current research project is in characterizing the immune system responses of turf grass species called creeping bentgrass and its response to a plant growth-promoting rhizobacteria, which is Bacillus subtilis. I'm trying to find how this beneficial bacteria could promote turfgrass performance and its resistance to certain turfgrass diseases.
Wonderful. And if you could have your dream research project, what would that be?
A dream project would be a one where I could extend my current research and further understand plant-to-plant interactions, plant-to-microbe interactions, and microbe-to-microbe interactions, and then use these beneficial interactions to improve plant performance and sustainability in agriculture. And it would be great if I could get opportunity to go to some exotic places to conduct some of this research.
Awesome. Well, if you'd like to get in touch with Charan about her work, we'll have her contact information in our show notes. Thank you so much for being on the show today, and best wishes on your future studies and career. Thank you.
Thank you.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.